It's 17 years since I gave my last lecture in Trinity College, so it brings back strange memories. I'm going to talk to you for 2 sessions about genetic genealogy, your last 2 sessions before the Christmas break, I'm told. I have put these notes up on my website with lots of live links to interesting websites. So all you really need to write down is the web address, pwaldron.info/TCD, and that's TCD all in uppercase. It is case sensitive. And hopefully this is being recorded, and I may put up a video of it on YouTube later on, which will be the screen and my voiceover. How many of you have sent your DNA off for analysis to a DNA company? How many have not? I'm surprised. I would have thought people signing up for a family history course would have done so already. So this is going to be starting from the basics, explaining why you should do it, and working up to some more advanced stuff by the end of next week's talk. I have about a dozen DNA kits from Family Tree DNA. If you get them from me, it saves you the $12.50 shipping fee. And they started their Thanksgiving sale this afternoon, so the basic family finder test is now only $49. I'm not a salesman for Family Tree DNA. For Y-DNA, which I'll come to later, they're much the best company. For autosomal DNA, they do not have the biggest database, which is a major drawback. Ancestry DNA, which has a huge marketing budget, as you've probably seen in television advertisements, now has over 10 million people in its autosomal DNA database. I'll explain the differences as we go along between autosomal and Y and the other components of DNA. And if you have any questions, feel free to shout and stop me. We may have the chance afterwards, if you want to sign up, to do the swabbing here and now, or I can give you the kit to take away. If you want to do it at the end of class, there's one rule, or wherever you do it, there's one rule: to get a pure DNA sample, no eating, drinking, smoking, chewing gum, or washing your teeth for an hour before you do your DNA swab, which you do with a little cotton bud that rubs the inside of your cheek and picks up some of your DNA from your cells. Can you all read that up at the back? So I'm going to start today, week 1 of this 2-week genetic genealogy section, talking about the context of using DNA in terms of what you've been learning so far in the family history course. And then I'm going to move on to some of the definitions, the different components of your DNA, the different inheritance paths down your family tree through which those components come to you, and then talk about what makes our DNA different and usable for family history research, the mutations that happen from time to time, and the recombination that happens when a child is conceived, how the parents, the fathers and mothers' DNA recombine to produce the child's DNA. Next week I'll go more into how to using the several DNA websites that are out there for DNA comparison, talk about the paradox of identity and anonymity. You're trying to identify your ancestors, and yet you're going to find a lot of your DNA matches are trying to remain anonymous. And I'll go through a number of case studies next week. So having talked about this with Fiona a couple of weeks ago, I said the bottom line is to point out to you, everyone in this room is the expert on his or her own family history. You have the family's oral traditions, which you have picked up at home as you grew up, or picked up as you visited relatives, or will be picking up this Christmas as you visit relatives and ask them questions you mightn't have thought to ask them at previous Christmas holidays. Somebody in your family, maybe yourself, has the family papers in the attic, in a butter box, in a biscuit tin, somewhere in the house, and they're one of your major sources as well. And you have the family DNA in every cell of your body, and that is becoming increasingly a more important source for your family history. And your task as a genealogist is to compile your family history by matching up those 3 records: the oral history, which is sometimes wrong, the family papers, where things might sometimes be exaggerated, and the family DNA, which sometimes doesn't match what you'd expect it to. We have what we call NPEs in DNA analysis. That started out as an abbreviation for something quite ridiculous, a non-paternal event, but it has now been reinterpreted as just meaning not the parent expected. Somewhere in your family tree, be prepared for a surprise. Typically the surprise will be an adoptee who might be a second or a third cousin who you've never heard of. Or it might be that the father isn't the father that you would expect from the family circumstances. So sometimes every piece of information reconciles neatly with each other. Sometimes there are conflicts, and it's the family historian's task to try and resolve the conflict, go out and look for other evidence and see which version of the story is true. So what's genetic genealogy? I said everybody these days probably uses Wikipedia, so I said I'd look at the Wikipedia definition, and it says genetic genealogy is the use of DNA testing in combination with traditional genealogical methods to infer relationships between individuals and to find ancestors. Well, I put the word testing in double quotes. In a past life, I was a statistician, and statisticians' job is to test hypotheses, and I guess family historians' job is also to test hypotheses. This is not what the DNA company does for you. Sending the DNA off to a lab is not the end, it's the beginning of the journey. You will use the information that you get back from the DNA lab to test your own hypotheses about your family information. Was John Smith, who lived in Nina in 1802, the John Smith whose son was married in America in 1840 or whatever? That sort of hypothesis. So you have tools provided by the DNA websites to help you test that sort of hypothesis. So the first thing I think anybody doing genealogy of any sort should do is to set up a database. How many of you have set up your family history in a database like Ancestral Quest or Roots Magic or Reunion or any of the others? A handful. How many have not? It's time to start, because you will be getting more and more information, and the only way you can make sense of it is with a database like this. This is my pedigree chart. There's me on the left-hand side, my parents, my grandparents, my great-grandparents, my great-great-grandparents. It's a very big database. You can search the database. That's picked out my matrilineal great-great-grandmother, Mary O'Neill, whose maiden name I still don't know after decades of searching. And I can scroll up and down the alphabetical list here. But I can also do clever things like defining field selections. And for every individual in this database that I have found in the DNA websites, I put a field in the notes which I've called GetMatch, because GetMatch is the best of the DNA websites. We'll come to that later. And I can say, highlight all the people who are in the DNA websites, and there are 1,826 of them that I have found and researched. And I can say, show me only those, and it will give me a list of only the people I found in the DNA websites. Or I can do something better. Let me go back to myself and say, how many of my DNA matches have I confirmed my relationship to? So I can say, show me all the people who are my ancestors, back 999 generations, that's the most it allows, and their descendants. So all my blood relatives with whom I might share DNA because I share an ancestor with them. And I've actually found 15,000 blood relatives, and 219 people in this database are both my blood relatives and on the DNA databases. It'll take you a while probably. I've been at it for years to build up to 219 DNA matches to whom you have traced a relationship. But that's the objective, and as the numbers get to that size, the only way of managing it is in one of these databases. And I've given you links there. I use Ancestral Quest today because I started using its predecessor way back in 1987. If I was starting today, I mightn't start with that one. Most of them have free downloadable trial versions with some limitations on the size of the database or some of the features. So try them all, see which you like. There's a link to a few more, and on Wikipedia, there's a whole load of examples listed with all their features. And of course, you can use some of the online genealogy websites to store your database. I don't recommend it. It's a lot slower unless you have lightning fast broadband down the part of the country where I live. We don't have lightning fast broadband yet. And of course, the other thing is you might commit yourself to one of these online websites in the cloud, and clouds have a habit of evaporating. You might have noticed the wonderful Ordnance Survey Maps website operated there evaporated there a few months ago. If you ever used Vodafone for your email, that evaporated a few years ago. So make sure you have a good backup strategy. If you put your life's effort into creating your family history, you don't want to disappear if your hard drive fails or if the website that you're using goes out of business. I upload my database from time to time to a website using software called TNG, the next generation. So I have a long introduction there, and you can click through to register. You have to register, I have to approve. It's not a public website, and anyone that I approve, which means anyone who's related to me, I'm happy to let them see it, can see the fruits of my research. And sometimes it's easier for me to look on the web if I'm out and about, or if I want to do something that Ancestral Quest doesn't do well. A lot of you probably want to write a family history that you can print out on paper and give to other family members to read. The database will do that for you. Or you can use the free-form notes in the database if you want to add a little more. Or you can just use the database as a guide when you're writing the book, the bestseller. So the other thing Fiona says she's been stressing to you is how genealogical records are created by the actions of our ancestors, and our own actions are creating genealogical records for our descendants who will come back looking at them in centuries to come. But we're the first generation still to be creating digital records of our own DNA. I would love to have the DNA of my parents, my grandparents, my great-grandparents, your children, grandchildren, great-grandchildren, nephews, grandnephews, whatever, in years to come, will be delighted that they will have access to your DNA in the online databases. The more people submit their DNA to the DNA databases, the more useful they become. There's a report on the database sizes that's updated every few months on a website called the DNA Geek. And there's your chart with the 5 major autosomal DNA comparison websites. The biggest one is Ancestry DNA, which claims to have over 10 million people, probably well over 10 million at this stage. The second one is 23andMe, which is not really a genealogy site. It's more for people who want to get predictions of their possible health problems out of their DNA. Then the other 3 are quite close together. The orange one is MyHeritage, which is a late arrival on the scene and has moved quickly into 3rd place. And then roughly equal, you have Family Tree DNA and GetMatch, which are for the serious, hardcore genetic genealogists. There are significant positive externalities, as an economist might say, to putting your DNA in a database. It might help somebody that isn't even related to you. It might prove that a particular DNA segment came to somebody else from his father's side rather than his mother's side, and that would prove that people on the other side are really on his mother's side. So there are ripples in the pond when you throw your DNA in there that benefit people other than yourself. Your descendants, as I said, will be eternally grateful to you for leaving them your DNA. So do unto them as you wish your ancestors had done for you. And if you're lucky enough to still have living parents or living grandparents, well, you have only half of your parents' DNA and only a quarter of your grandparents' DNA, so go out and swab them as well. Deciding not to use your DNA for genealogy, some people say, is the equivalent of saying, I'm not going to use census returns for my genealogy. It's as important as using the census returns. As I said, sometimes the DNA refutes the paper evidence, but more often than not it confirms it. But the one warning is, it has let the genie out of the bottle as regards secret adoptions and fosterings. If there is a family secret in your family and you want to keep it a secret, the only way of keeping it a secret is to keep your DNA out of the databases and keep all your relatives' DNA out of the databases as well. I rang a man that I know recently, and I had found an adoptee who I figured was about his second cousin, and I thought I might be able to persuade him to let his DNA be used to help her confirm her ancestry. And he said, I'm not putting my DNA in any public database. I might want to rob a bank someday, and I'd be caught if my DNA was out there. 2 days later, I had a phone call from his brother, who'd already submitted his DNA to 23andMe. So if one brother robbed the bank, the other brother's DNA could be found to catch him straight away. Actually, there is only one of these database websites that says that it can be used for the investigation of crimes, and it is limited to murders, rapes, and serious violent crimes. And the others say, we do not allow our database to be used for criminal investigation. But it is possible to pick up a DNA sample at a crime scene and turn it into a file that can be uploaded to at least GetMatch to one of the genetic genealogy websites, and that has been done very successfully for the first time this year. You heard the story of the Golden State Killer in California, a man who had been a mass murderer, had been on the run for decades, and he had left DNA samples at various crime scenes, and using getmatch.com, a lady called Barbara Ray Venter was able to narrow down the list of suspects to a handful of people, and eventually to just one possible suspect. And then the law enforcement agencies picked up a discarded coffee cup and got DNA off the discarded coffee cup, and lo and behold, as she had predicted, it exactly matched the DNA from the crime scene. So he has now been charged. But without the discarded DNA off the coffee cup exactly matching the DNA from the crime scene, he couldn't be charged because it wasn't certain that it wasn't some other family member who had a familial DNA match to the crime scene DNA that was the criminal. So I'm going to talk in these two sessions about the methods to get the best out of your DNA as a genealogical source. The objectives are to interrogate and to extract the relevant evidence from your DNA, as you do with any other source. If you have an old document, keep reading it and rereading it, and you'll pick up another clue. I had a document from a probate case that I read at the weekend that I've had for years, and it said the heirs are blah, 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 and John O'Dea of County Clare, Ireland. And they were in little family groups, the heirs, so I figured this John O'Dea of County Clare, Ireland was on his own. He must be an only child. And then I looked at my family tree, and there was a John O'Dea who looked like an only child, and his descendants were close DNA matches to the descendants of the other heirs in this inheritance case, and suddenly I knew exactly how they all fitted together because of that one little clue in a probate document that I'd had for years that I hadn't reread recently. So your DNA will give you hints to go back and look at other sources, and vice versa. The other sources will give you hints to explain the DNA matches. DNA will also link you to genetic communities. That's a phrase that Ancestry DNA actually uses. You will find groups of DNA matches that you won't know the common ancestor for. The common ancestor will be somewhere just on the other side of that brick wall that you're trying to break down in your research. And eventually, as you get together and collaborate and chip away at the brick wall, you will eventually find out how to go back another generation. It is well known that DNA variations are closely correlated with geography. People who live close together have similar DNA. That's why you see these advertisements selling you estimated ethnicity percentages. You're 97% Irish or 33% Italian or whatever. Don't fall into the trap of assuming the opposite. If you have people living in place A and people living in place B who share DNA, that does not mean that their common ancestor did not live in place C and have descendants who migrated to two different places. I've heard so many tales of how the fact that people in Ireland and people in the Basque Country share DNA, hence the Irish came from the Basque Country. DNA alone does not tell you that. It tells you that the people in those two areas had common ancestors. It doesn't tell you anything without additional evidence about where the common ancestor lived. And I heard a good phrase recently. The DNA companies, I was out on a tour in, I can't even remember the name of the company now, in Cherrywood, Genomics Ireland or Genomics Medicine Ireland or something. And they've come up with this wonderful phrase. They're looking for the GAA people, people with geographically anchored ancestry. In other words, people who are certain that all their grandparents, great-grandparents, and great-great-grandparents lived in a particular small area, and they can assign genetic variants that are found in that person to that geographic area, and then say somebody else who has that genetic variant has relatives who lived in that area, and if the match is close enough, maybe ancestors who lived in that area. But again, the possibility is that the common ancestor lived somewhere else and migration went in two different directions. So when we look at DNA matches, we talk about all the usual suspects are in there, all the people with ancestry in the same area that we know are related if we could only go back a generation or two further. And there's also a great community of people to help you interpret your DNA results. Family Tree DNA, one of the big companies, hosts DNA projects. I co-administer a number of these. I just put in the list there. My grandmother came from County Clare, so I decided to take on the County Clare project, the Clare Roots DNA project. It's now got way too big. It's got 1,177 members. It has a web page on the Family Tree DNA website. Anybody who has an ancestor in County Clare is welcome to join it. And it provides, for example, an activity feed or a discussion board where people can ask each other questions, or they can ask me as administrator to use some of the administrator's tools to tell them a little bit more. Activity feed isn't going to load, but we don't need it. I also work on 5 different surname projects: Clancy, Durkin, Marnin, McNamara, and O'D. You will find a project for your surname unless it's very rare. If you have a very rare surname and there's no project there for it, well, you can apply and set one up and be the administrator yourself. And there are also what they call haplogroup-based projects for men with similar Y-DNA signatures, so groups of surnames with a common ancestor before the introduction of surnames. So, for example, L226 is also known as Irish type 3, but a better name for it is the Dalcashian DNA signature. The Dalcash were the people of County Clare, including Brian Boru, the High King of Ireland, killed 1,004 years ago at the Battle of Clontarf, from whom all O'Briens claim descent. In fact, very few O'Briens are really descended from Brian Boru. A common surname like that tends to have many different genetic origins. But the old Irish annals will tell you this is the origin of all these County Clare surnames, and they all trace back to Cos, who lived in the 5th century or thereabouts. And it turns out, yes, there are similarities between the Y chromosome of people with all those surnames. So they group together in a haplogroup project to discuss the evolution of the surnames in the 1st millennium and the start of the 2nd millennium AD. We have conferences. Genetic Genealogy Ireland has been going every year. Has anyone been to that? Nobody? 1 or 2? Part of Back to Our Past in the RDS every October. And this year, Back to Our Past and Genetic Genealogy Ireland started a Belfast version. So that will be on the 15th and 16th of February in Belfast. And then it'll be back in Dublin, I hope, next October, but the dates haven't been announced yet. You should all join the International Society of Genetic Genealogy. It's free to join. There's a link there to the application form. And it has a Facebook group. It has a wiki, like Wikipedia, with lots of useful information on genetic genealogy. And the Irish branch of ISOG campaigns and various things, like we put together a submission to the Minister for Children on the Adoption Information and Tracing Bill, which has been before the Oireachtas for years and does not make a single mention of the use of DNA to help adoptees to find their birth parents. So it shows how far the government is behind the recent scientific progress in this field. There's a list of some of the other Facebook groups. ISOG has one, Genetic Genealogy Ireland has one. There's a Genetic Genealogy Tips and Techniques group. There are dozens of those, but that one run by an American called Blaine Bettinger, I think, is probably the most sensible because he's a good moderator. So, down to the basics. What is DNA? It's a big word, deoxyribonucleic acid. You don't need to know it or remember it. Nobody does. The important thing is that it is made up of chromosomes and mitochondria made up of molecules of 4 different chemical compounds called nucleotides. They're adenine, cytosine, guanine, and thymine. Again, you don't need to know those words. All you need to know is you spit into a tube for ancestry, or you swab your cheek for family tree DNA or whatever. You send your DNA sample off to a lab, and it is turned into a big computer data file of the letters A, C, G, and T, representing your personal unique DNA. And you can download that data file from the lab website and upload it to several other DNA comparison websites which have different groups of people who've submitted their DNA and different tools for analyzing the DNA. But we all get our DNA from our parents. If it was all deterministic, well, we'd all have the same DNA, but there's a little bit of random variation in there, which means we all have subtly different DNA. About 99-point-something% of every human's DNA is the same as every other human. It's the 0-point-something% that's different that is useful for genealogy. I think about 95% of our DNA is shared with the apes. So we're just looking at a tiny little bit, which is good because there's a huge amount of DNA, and even the tiny little bit that varies is still an awful lot of data to analyze. So we all start out as a sperm fertilizing an egg. The sperm has either a Y chromosome or an X chromosome. If it has a Y chromosome, it ends up as a male child. If it has an X chromosome, it ends up as a female child. The father, through the sperm, also provides 22 paternal autosomal chromosomes, which match up with 22 maternal autosomal chromosomes which come in the egg from the mother. The egg also brings an X chromosome. For the woman, for a female child, that will be a second X chromosome, one from each parent. For a male child, there'll be a Y chromosome from the father with an X from the mother. And the egg also has what they're called the mitochondria, a small piece of DNA in the nucleus of the cell, which passes from the mother's mother, and so on. Each of these components has its own inheritance path. So if we go back to my pedigree chart, my Y chromosome came from my father, his Y chromosome came from his father, his Y chromosome came from his father, and so on back along. The Y chromosome comes down the top line of the pedigree chart the same way that the surname does. So the Y chromosome is great for one-name studies and surname studies. The mitochondrial DNA, on the other hand, I got from my mother, who got it from her mother, who got it from her mother, who got it from her mother. So it comes along the bottom line of the pedigree chart, and there's a couple of problems with this. From a genealogical point of view, normally along that line, the surname is different for every generation. In my case, my grandparents were both Durkins on my mother's side, so there wasn't quite as many changes of surname as there might be. The other problem is the mitochondria is small and slow mutating, so compared to the Y DNA, you may have to go back many generations to find the common ancestor, even if you have a perfect match on the mitochondrial DNA. The autosomal DNA, 22 chromosomes from my father, 22 from my mother, exactly half from each. On average, a quarter of that comes from each of my 4 grandparents. On average, 1/8 of the autosomal DNA comes from each of the 8 great-grandparents. On average, 1/16 from each of the great-great-grandparents. Not exactly 1/16, and eventually as you go back through the generations, there will be ancestors from whom you will inherit no autosomal DNA, because there's recombination and those percentages vary a little bit. So if you compare your autosomal DNA to a cousin's autosomal DNA, if the relationship is closer than third cousin, you're guaranteed to find that you share autosomal DNA and that you will be returned as matches to each other. At the third cousin level, you start to have a chance that you've inherited different bits of DNA from your shared great-great-grandparents, and there's a small probability, maybe about 10%, that 2 third cousins will not match with each other. If you compare the autosomal DNA of 2 fourth cousins, the chances that you've all inherited different bits of the shared great-great-great-grandparents' DNA increase to about 50%. When you get out to fifth cousins, there's only about a 10% chance that two fifth cousins will show up as DNA matches. We have an awful lot of fifth cousins, and sixth cousins, and seventh cousins. And if only a couple of percent of your sixth and seventh cousins share DNA with you, that's still an awful lot of DNA matches to try and work out the relationships with. Sorry, that wasn't supposed to happen. So, to summarize, only men have a Y chromosome, comes down the same line as the surname from the father, father's father, and so on. Same male line as grants of arms, peerages, other things in a patrilineal society. X chromosome is messy because a man has one, a woman has two. It can come down, or bits of it can come down from any ancestor as long as you're not going through two consecutive males on your way back to that ancestor. And there's a chart that you can look at just to explain that. Here's a fan chart with the father on the left for a man, the mother on the right. The man gets only one X chromosome from his mother, who got bits of her X chromosomes from both parents, but her father only got his from his mother, and any of the coloured squares can give a bit of your X DNA, and the white squares don't give you any of it. We usually only use the X DNA for targeted research questions. You might be looking at two men and wondering whether they were brothers. If they were brothers, they will share X DNA from their mothers, and their descendants may share X DNA. So that's a good signal that they are brothers. If they don't share X DNA after many generations, it doesn't necessarily disprove your theory, but sharing it will prove they're brothers rather than first cousins. Because first cousins will have different, with the same surname, will have different mothers and different X chromosomes. The autosomal chromosomes come 50% from each parent, on average 25% from each grandparent, and so on. I found a case recently where that 25% average didn't apply, an extreme outlier where a girl had 35% of her DNA from one paternal grandparent and only 15% from the other. So it can vary 25% plus or minus, but 15 and 35 is very extreme. Two siblings each get half of their parents' DNA, their parents' autosomal DNA. They do not get the same half unless they're identical twins, unless the egg splits after it's been fertilized. So don't assume just because your brother or your sister has submitted their DNA that your DNA is not of value. You will have another quarter of your parents' DNA that your brother or sister doesn't have, and there'll still be a quarter that neither of you has. The same thing with the X chromosome. Two siblings get half of their mother's X DNA, but not the same half. Two sisters get their father's single X chromosome exactly, and the mitochondria comes down the female line. So I said I'd summarize all this in another table, because it's something that I've talked about many times, and at the end of the class it still hasn't sunk in. From your father only, you get your Y chromosome, provided that you're male. Equally from both parents, you get your autosomal chromosomes, whether you're male or female. The X chromosome, if you're male, you get one. If you're female, you get two. That's a bit messy. From your mother only, you get your mitochondrial DNA, whether you're male or female. So if we're using this for genealogy, rapidly the autosomal DNA, the 22 pairs of chromosomes that come equally from both sides, has become the cheapest to analyze and the most widely used in genealogy. The Y chromosome was being used maybe 10 years before anybody started using autosomal DNA for surname studies and one-name studies. But there have been vast scientific advances in the analysis of the Y chromosome also in the last couple of years. And the other two, the mitochondrial and the xDNA, are typically only used for specialized problems. They're not used for fishing trips. We say you go on a fishing trip by throwing your DNA out into the ocean. That's the online DNA database to see what you catch. And you do that with your Y DNA to try and catch other men with the same surname. And you do that with your autosomal DNA to try and catch your first, second, third, fourth cousins on all sides of your family. So I said we'd all be very uninteresting if our DNA was passed on unchanged from father to son. What makes it interesting and easy to use for genealogy are the mutations and the recombinations. Mutation just means a transcription error. A parent might have a letter A in one location and the child might have a letter C instead of the same letter that the parent had. And there are some locations where these type of mutations happen very frequently, and that's good for the criminal investigation people, because if you look at those frequently mutating locations, two people who match on a large number of those, or two samples which match on a large number of those, have to come from the same person. So the people using DNA to uniquely identify a criminal are looking at different locations than those of us using DNA to identify our close relations. We want to look at mutations that occur less frequently, only once in many generations or once in the history of mankind, to match up people who are closely related. So that's another reason why the genetic genealogy databases historically have not been much good for criminal investigation, because they can only say, well, this guy is closely related to the guy who committed the crime. They can't say he is the guy who committed the crime. So there's two types of mutation that are used. The STR is short for short tandem repeat, and in your DNA you will find that there are sequences like this CCTG, which repeat over and over again in succession. In that case, it's repeated 7 times in a row, but that mutates frequently, and the next generation it might occur 8 times. There might be an extra CCTG stuck in, or one might drop out. Typically it passes on unchanged, but some of them mutate very frequently, some very rarely, and they were what were first used on the Y chromosome to identify closely related men, looking at these sequences, these STRs, and saying the men with 7 repeats of that STR is closely related. The other man with 7 repeats and the guy with 9 repeats is more distantly related. The second bit of jargon you need is the SNP. That's short for single nucleotide polymorphism. Again, we just use the abbreviations. You don't need to remember the big words. And that is a location where 2 different letters have been observed in different individuals. Some people have an A, some people have a C. Very often it's a once in the history of mankind mutation, so that most people have the A, and everyone with the C is descended from the first person who ever had that mutation. And in the case of the autosomal DNA, a mutation like that is going to spread and spread, because on average every person has 2 or more children, 4 or more grandchildren, 16 or more great-grandchildren, and that helps the SNP to spread. And the same thing in the Y chromosome. The SNP will descend like the surname from generation to generation. So let's talk a little bit about how the Y DNA has been analysed. It passes virtually unchanged from father to son, but men have been passing on Y chromosomes to their sons for hundreds of thousands of years now, and even if there are very rare mutations, after hundreds of thousands of years we have ended up with a wide variety of Y DNA signatures. The analysis started off about 20 years ago looking at 12 STR mutations, and they were able to find a lot of patterns just with those 12 mutations. Now they're up to standard 111 STR locations. Family Tree DNA is really the only company on the scene for Y DNA comparison, and as I said, it runs surname projects. So this is one of the projects I co-administer. It's a bit small there on the screen, but you can see the general pattern. There are about 20 members in the project. The first column is their kit number. The second column is the surname. You'll see most of them are Durkins, spelled 3 different ways, because the spellings mutate in the same way that the DNA mutates. Some people spell it with a C, some with a K, some spell it with an A, some spell it with an I. Most of the Durkins have similar Y DNA signatures, and they've been grouped together, but there are 2 outliers down here whose Y DNA signatures don't match. That means there has been a surname DNA switch at some point in the past. a son got a different surname from his father. He might have taken his surname from a stepfather. He might have taken his mother's surname rather than his father's for some reason. He might have been fostered or adopted and taken a foster father or an adoptive father's surname. There are many reasons for surname DNA switches. And sometimes you can tell when two men with the same surname don't match. Well, it wasn't a surname DNA switch because the DNA signatures are so different that perhaps two different men adopted the same surname 1,000 years ago, and there's no genetic connection at all. So with a common surname like Smith or Miller that comes from an occupation, you can well imagine that there might be 27 different Smiths around the world, blacksmiths around the world, who have founded 27 unrelated dynasties with the Smith surname. So it may be the common ancestor lived before surnames, rather than that there was a surname DNA switch in the last 1,000 years or so since when surnames have been used. Here in Ireland, we're supposed to have the oldest surnames in the world. I mentioned Brian Boru already. They say that when Brian Boru's descendants adopted the O'Brien surname, that was possibly the first use of a hereditary surname anywhere in the world. There are other countries. Iceland, I think, still doesn't have surnames that go from father to son. Some countries have only had them for a couple of hundred years. We've had them for nearly 1,000 years. The third column here is very important to fill in. You're asked to give your most distant known paternal ancestor name. As you can see, half a dozen people have left them blank, or one of them has even just said unknown, or one has just given the surname Durkin. But some of them, you have 50 characters here to fill in all you know about your most distant male line ancestor. So, well, I'm down here. My mother was a Durkin, so I'm grouped with the people with other surnames who are in the project because we have a Durkin ancestor not in the male line. So all I could fit in is Thomas Waldron from about 1825-26 in Roscommon and died in 1902 in Limerick. So try to fit in as much as you can there to give people an idea of where your ancestor lived and when your ancestor lived. The next column gives you details of the SNP mutations that are either predicted from the STR mutations or tested for directly. I'll come back to those maybe the next day. And then you have 111 columns over to the right. These are the 111 STR markers that are used to compare people and to measure a genetic distance. The genetic distance between two men is essentially the number of differences between one row of numbers in this table and the next row of numbers. So you see this first group of Durkins have all the same values. The red numbers on a red background are one or more above the mode. The numbers on a blue background are one or more below the mode. So the colored bits are where they differ from each other. So down here where you have three people with three different surnames and no male line connection, there's lots of color because we have lots of differences. Up in the top group where they're all Durkins and they're all related to each other, there's very little color because there are very few differences. And you can see here's the 37th column, 37 markers is the entry level now that most people go for. And then you have some blanks because some of them have only paid for the first 37 markers to be analysed. Then you have another batch of 30. These ones have only paid for 67. And then the real addicts pay for all 111 to be analysed. And again, you see an odd difference because since the Durkin surname was adopted several hundred years ago, there have been occasional mutations, but most people with the surname Durkin match on most of the markers. Now we have a thing called the Big Y 500, so they've actually increased the available number of STR mutations that they analysed to over 500, but they're not actually using those yet for calculating genetic distances or matching men together. Genetic distance is the number of differences between those STR signatures. So in the old days, you might find somebody with a 0 genetic distance, but you're only looking at 12 markers. That tells you you have a common ancestor sometime in the last couple of 1,000 years, probably. Nowadays, looking at 111 markers, a genetic distance of 9 out of 111 will typically be somebody with the same surname with a common ancestor within the last 400 or 500 years. The smaller the genetic difference, The smaller the genetic distance, the closer the relationship. When we come on to autosomal DNA, we will look at genetic length, and in that case it's the opposite. The bigger number is the closer relationship. But when you're comparing strings of STRs, you want them to be close together, and a zero distance is the closest relationship. The correlation is not perfect. You can have two brothers and one has had a mutation that the father didn't have, so there's two brothers with a genetic distance of 1 between them. Or you can have two men with the same surname who can trace their pedigrees back 6 or 7 generations without meeting a common ancestor, and they can have 0 genetic distance because there has been no mutation in the last 6 or 7 generations in either branch. And that's why people move up from 37 to 67 to 111 markers. You're more likely to have a mutation to distinguish branches if you look at more markers. Spellings mutate, I said that already. And we can use these mutations to build a haplotree. This is how it started about 20 years ago. They didn't even need all the letters in the alphabet to describe all the different Y-DNA signatures that had been discovered. They only went up to the letter R, so that's about 18 letters. And that's the way they figured out they were all related to each other. And they all go back to a man, an unknown man, 150, 250,000 years ago, who's been christened the Y Adam. He's the only man alive at his time who has male line descendants still alive today. There were many other men around 250,000 years ago, but as you probably know from tracing surnames in your own family, it doesn't take long for a surname to die out if a man has only daughters, or if his sons don't marry, or if they go into the priesthood and become celibate for that reason or whatever. And over 250,000 years, there have been a lot of male lines that have been daughtered out, and there's only one still alive. So, not the same as Adam in the Bible, who was the only man alive at the time. Y Adam is the only man alive at his time who has direct male line descendants still living today. Now, what does that haplotree look like after 20 years of research? Family Tree DNA have recently published a public version of the Y-DNA haplotree. So it starts out with those 18 branches from 20 years ago, but now there are 16,000 branches and 160,000 confirmed SNP mutations. So there's the branch for haplogroup OR, which is the most common haplogroup in Ireland, and you can view it by countries, by surnames, and by variants. So here by countries, can we see an Irish flag in there anywhere? 4 Irish people out of 19 in that branch of the haplotree. And I'm not sure why it's only showing 19 flags. Oh, they're the people who probably have that mutation, but none of the later mutations that have been discovered to go with it. There's 8,560 in that branch, of which 7,313 are in that branch. And you can keep clicking down, opening out the branches. And we go on with the most common one each time, 7,000 people in that branch. And little branches branch off with fewer and fewer members. And it's one enormous tree with hundreds of thousands of people in it now, and hundreds of thousands of mutations. So it's grown from the other little picture in 20 years to that. Mitochondrial, I should finish up now in a couple of minutes, so maybe we leave recombination for the next day, or just give you time for a few questions. Mitochondrial mutations are rarer because the mitochondria is much smaller than the Y chromosome, and as I said, it's difficult to find a common ancestor if you even have a perfect match, but there is a mitochondrial haplogroup tree, and there was a book written called The 7 Daughters of Eve, talking about the 7 main branches of the mitochondrial haplotree some years ago. The autosomal DNA mutations mean that, as I said, 99% plus of the autosomal DNA is the same for everyone. So what the DNA companies look at is about 0.02% of the locations on the autosomal chromosomes. They're what make us different from each other. They're what vary. I think they probably could do a lot more on the comparisons, because they give the same weight to a rare mutation that only 1% of the population has than they give to a very common mutation that half the population has. So what is recombination? We'll finish with this and I can go over it again the next day. So let me show you this little stylized example. Blue for the father, red for the mother. This is the string of letters in, say, the father's chromosome 7. This is the string of letters in the mother's chromosome 7, or in my paternal chromosome 7 and my maternal chromosome 7. So I have 2 chromosome 7s, one from my father, one from my mother. When a sperm or an egg is created, it takes half the paternal chromosome and half the maternal chromosome, and it crosses back and forth. So the combined or recombined chromosome will start out with a blue string taken from the paternal side, then it'll cross over and take a red string, which is a copy of the maternal side, then it'll cross back and take another string of blue letters from the father's side, and then it'll cross back and take another string of red letters from the mother's side. So I've done a stylized example there where about every 20 letters there's a crossover. In fact, crossovers on average happen about once every 19,000 letters. So there's an awful lot of letters out there. If you print out your genome on paper, it'll fill half this room. There are billions of letters in it. But basically what that means is every sperm and every egg is unique because the crossovers happen in different places. And every human being is therefore unique. And we have to try and patch these things together to find matches. The recombination rates vary as you move along the chromosomes. So there is a unit called the centimorgan, a unit of genetic length, to tell you how closely 2 people are related. So in this case, the bigger the number, the closer the relationship. The definition is that we expect one of those crossover events per generation in 100 centimorgans. So on average, you get 1 recombination per 19,000 observed letters. And we have a correlation between the centimorgan length of the shared DNA between 2 people and the relationship. So I'll just end with 2 graphs to give you a rough idea of what sort of numbers you're going to be looking at when you do autosomal comparison. A parent-child will share about 3,500. A parent-grandchild will share about 1,700, but it can vary from 1,100 up to 2,300. An aunt or uncle you'll share about 1,700 with. A first cousin you'll share about 870. A second cousin about 200. The third cousin about 74. Fourth and fifth cousins, very small numbers. So you will get a feel as you look at your matches for what number relates to what relationship. But a third cousin can vary anything from 0 to 217, a fourth cousin 46 to 515. So somebody who you share 150 with, you don't know whether it's a second cousin or a third cousin, but if it's 150, it's certainly not a first cousin. It's too little to be a first cousin, and it's certainly not a fourth cousin. It's too much to be a fourth cousin. And that's one type of chart. The other chart I look at turns it into probability terms. I don't know whether you can read those numbers up there. I can blow it right up for you. It divides up the possible relationships into 10 groups. There's the full sibling relationship. There's the grandparent-grandchild relationship, which shares the same expected average amount of DNA as a half-sibling relationship or as an uncle or aunt to niece or nephew relationship. Then there's the first cousin category, which shares the same amount of DNA as a great-uncle, great-nephew comparison. First cousin once removed will share the same expected amount of DNA as, say, a half-first cousin. So there's these 9 different categories. If you get a number over 2,400, you're guaranteed it's in the full sibling category. If you get a number between 1,550 and 2,000, you're guaranteed it's in the half-sibling category. But if it's between 2,000 and 2,400, it could be a full sibling with whom you share less than average. Because recombination is random, it doesn't give you an exact value. Or between 2,000 and 2,400, it could be a half-sibling sharing more than the average. The same thing between 1,000 and 2,000, it could be a half-sibling sharing less than average, or it could be a first cousin sharing more than average. And when you get down to the bottom, you really can't tell the difference between a second cousin once removed and a distant cousin, or between a second cousin and a fourth cousin. But there are probabilities here. So if your shared DNA is 130, there's about a 50% chance you're looking at a second cousin once removed, but there's 14% chance it's closer, and there's 36% chance it's more distant. So I will show you the actual DNA comparison websites next week, and how to put those numbers into practice. And I will work through some case studies with you. I'm happy to answer any questions anybody might have, and if anybody who hasn't done a DNA test and has been persuaded to do the family tree DNA test, I can give you the kit. Or if you want to go into the Ancestry DNA database, which is much bigger, you can order that online. And it's $59, I think. They have sales for Thanksgiving and the holidays, as they call it in America, and they've started already. So now is a good time to sign up.